is a part of a cell called dendrite. It's a dendritic segment, and you see all these little thorny projections. They are the places where the connections with the other neurons are made. And today I'm going to tell you a story of how communication between neurons is the key to really remember different important information or different types of information. Now, before we start, or before I start talking about the mechanisms of memory formation, let me tell you a little bit more about myself. Dr. Stefan gave a nice introduction. Yes, I'm Anant Jain. I'm currently a postdoctoral scholar at Max Planck Institute here at Jupiter, and I'm in Dr. Rohey Yasuda's lab. I joined as a postdoc in September 2019, I think. So it's been more than a year, and so far I'm really enjoying it. It's a great environment to do science in a stress-free manner. And it has an excellent team of scientists and excellent technological infrastructure. So far it's been amazing. Now, how did I reach to Max Planck? So I come originally from a small city called Bhopal in India. Now, Bhopal is not known for famous reasons. I don't know if any of you heard about this. Bhopal is known for the world's second worst industrial disaster called the Bhopal gas tragedy. It happened in 1982, I think 2nd December 1982. And this was like worst after the Chernobyl, that is more popular industrial disaster. And even after many decades this event happened, there are still a lot of genetic defects and a lot of problems, environmental problems that are still seen in the city, you know. And so seeing some of these genetic mutations that occur due to the release of this methyl isocyanide gas really motivated me to learn more about the genes. And so when I stepped out of Bhopal for my undergraduate, I went to Chennai, which is like a city in the southernmost part of the country in India, very close to Sri Lanka. And here I did my undergraduate in the field of genetic engineering. So literally I was studying how the genes can be manipulated and engineered in various different ways, and how that can be used to our advantage. For my final year of undergraduate, I went to Boston. So this was my first international trip, and my first real experience doing research was at Harvard Medical School. So for my final year, I worked in Dr. Anna Krichevsky's lab and trying to understand a glioblastoma and how is this the most aggressive form of brain tumor. And really here I started appreciating how brain is such a complex system and how much we don't know about it. So I started, I did an internship here. I did my final year of bachelor thesis here. And after that, I thought that maybe I should do a PhD in neuroscience, and I applied to some top schools. I thought that, you know, after doing, spending some time at Harvard, I might get the opportunity. So I applied to like 4 or 5 top schools that year, and I got rejected. And I was like, oh, I didn't even get an interview call. And the reason was I needed some more research experience in the neuroscience field. And so I started applying to different labs all over, and I got an opportunity in a totally different part of the world, in Japan, Okinawa. So my next stop was in this Okinawa Institute of Science and Technology, where I was working in Dr. Eric D. Shooter's lab and really making these computational models of how the synapses can change in this brain region called cerebellum, which is important for our motor memory. And this is, and retrospectively, I think this was where, you know, doing research on the beaches of Okinawa, I fell in love with the current research that I'm doing. So after a year of internship here in Okinawa, I started applying for PhD programs again. And this time I got into the PhD program at Northwestern, like Stefan mentioned, and I came back to Chicago. So now this university has one of the biggest interdisciplinary neuroscience programs, and I was really excited to start my PhD. And I think this was one of the best learning experiences in my life. I did my PhD under Dr. Catherine Gui. She was my advisor, and I defended my thesis in 2019, and soon after Chicago, I started finding it too cold, so I came to a warmer place, Florida. And so here in Florida, and that's how I reached. Now, when I started from Bhopal, I never really imagined or never even dreamt about this kind of life journey. But this is something that I say to the youngsters: don't try to chalk out your entire lifetime. Do what you're really passionate about, do what you really enjoy, and just look out for opportunities and grab the best opportunities at any given time, and you'll really have a good life. All right. So this is a little bit about myself. So now let's move on to the talk. Now, before we start talking about the mechanisms of memory formation, I want to emphasize on the fact of why is it so important to study memory? And the reason is mental health. I think that mental health could be the biggest medical challenge of 21st century. These are data from the National Institute of Mental Health. We know today that every 1 in 5 US adult experience mental illness. Every 1 in 20 US adult experience a serious mental illness that really require a lot of money and grants to treat them. This is not only true in aged members, 17% of the youth really experience some form of mental health disorder. And if you break the different mental disorders, 19% of the US population undergoes some kind of anxiety disorder, some form of anxiety. 8% go in depression, some form of depression. 4% undergoes some kind of post-traumatic stress disorder. So there are all these mental health problems, and these numbers are going to get dramatically high and more severe in the next decade. And we also know that many of these mental illnesses, or the mental illnesses like stress and anxiety or depression, substance use, commonly affect one faculty of the brain, or one function of the brain, which is our capacity to learn and form memories. And this can also get a little bit more serious with age, or even in youngsters. We can have problems like serious memory loss, like dementia, and this is a component of severe neurological disorders like Alzheimer's or Parkinson's. And so this really points to the fact that understanding memory is really important to start delving into these mental health issues. One more point is, today, we can't treat memory loss with the same efficiency and efficacy as we treat bacterial infections or blood clots or high blood pressure, right? And the reason is we still don't understand memory loss to the same extent as we understand some of these phenomena, how it happens. And so my talk today will focus on the advancements scientists have made to really understand memory from starting from a theoretical perspective to the theories that have been developed to some of the treatment options, or some of the things that we can use to develop therapeutic strategies. So I'll talk about some of the advancements and what are the future directions. Now my talk is divided into 3 components. The first part of the talk will describe the different types of memories. Now this is a psychological perspective which will help us to just come on the same page of what we really understand by memories. Then we learn about how do neurons communicate. So, and why does this neuronal communication is important for memory formation. And the last one will be how is information processing takes place in a synapse. Now I'll explain the synapse and we'll get to that. Now, I know it's 7 o'clock. I know our brains are tired, so I'll try to make it less like a seminar or a lecture, and just try to tell you some more stories and the cool experiments that the scientists have done, right? But if you feel at any point that things are going too boring, just somehow tell me, and I'll go faster in that section. All right, so let's start with the first section of the different types of memory that our brain forms. Now, memory can be defined as faculty by which the brain encodes, stores, and retrieves information. And all these 3 components are equally important to really define what is a memory. If you are storing a lot of information, but if you are not able to retrieve or recall that information, that's not a memory anymore. And so all these 3 components are equally important. Now, memory can be categorized in many different ways. One way to categorize memory is by how long you can store a particular kind of memory. So for example, you can categorize them as sensory memory, short-term memory, or long-term memory, depending on how long it stays in the brain. Sensory memory, for example, lasts for less than 1 second. And these are literally the sensory information that you receive every single moment in your life, right? This is things that you are seeing while you're driving. You see a number plate, you see a billboard, and these are the information that's coming through your eyes, called the iconic memory. And you can have information about or the auditory information that's coming in constantly, which is the haptic memory, or the memory determined by your ears, or you can have the touch memory. All these different senses are constantly bringing in the information and creating these sensory memories, which last for less than a second. There are also other senses that I'm not mentioned. For humans, these three overpowers the other senses. But in other species, for example, odor might be more important than vision. All right, so we have these sensory memories that comes in. And then the other type of memory is the short-term memory, also called the working memory. Now, working here, the brain starts to combine the sensory information and starts making the assessment whether this information is important or not. And it starts associating it with already existing information. Now, this is a memory that lasts for a few seconds to a few minutes, and it has a very limited capacity. Initial behavioral scientists estimated that we can store up to 7 elements in this memory, and that's why the original phone numbers are also 7 digits long. Now, working memory is actually a very interesting concept, and people have developed a lot of interesting behavioral tests. And a simple example of that I can give you is, if I ask you to calculate 12 multiplied by 5, what you do is you got this auditory information, but now you'll be working in the brain to do some calculation, and you'll give me some answer, right? And that's what basically the working memory is doing. Literally, it is containing some information for a little bit for helping us to solve a particular task on our day-to-day basis. Now, understanding working memory, I'll give you another exercise. If you close your eyes, if I can ask everyone to close their eyes and then tell me the number of windows that are there in your house. Okay, so I'll just give a few seconds. Maybe you can just close your eyes and tell me the number of windows in your house. Now, what most of you did was that you closed your eyes and then you created a visual spatial map of your house. You physically located in one part of the house, and then you literally went from one window to the other and started counting, right? And that is what working memory is. It's basically combining your different sensory information with the already existing information and trying to solve a particular day-to-day task. Another interesting task is the Stroop test, which is also about the working memory. Now, I don't know if people have seen this before, but this task has two parts. The first part will ask you to really read out the letters and forget about what color they are. So that's easy, right? So you can read red, yellow, blue, green, green, yellow, blue, red, blue, red, blue, green, yellow, and so on. The second part of the task will ask you to tell the color of the word and not really read the word. Now, this gets a bit challenging. The first line is easy because it's the same: red, yellow, blue, green, green, yellow, blue. Yellow, blue, red, green, yellow, red, green, and so you should try it out too. It can be found online, but you can see that there is a little bit more difficulty in really telling the colors than just reading out the words, right? And this tells us how the information is combined in different ways by this brain region that is involved in working memory. And so there are interesting behavioral tasks like that to really study working memory. And this is a short-term memory. Now, the last one is the long-term memory, which is the memory that lasts for many minutes to up to many years. And in contrast to working memory, it has infinite capacity almost. Now, long-term memory can also be of many types. The first type could be that it is an explicit or a declarative memory. Now, this is a memory which is what is actually considered as a memory. Like if I'll ask you to tell me some capitals of the country, for example, what's the capital of France or US or India, you'll try to consciously recall that information. Or if I'll ask you, what did you do for your 18th birthday or your first marriage anniversary, you will consciously try to recall that information. And so this conscious recalling or retrieval of memory is the explicit or the declarative memory. However, there are some other non-declarative types of memory where you are not consciously recalling. So, for example, if you are learning how to drive, if you're learning how to swim, if you're learning how to play a piano, those are all your skills, right? And the skills, and while you're performing, while you're driving, you're not consciously thinking that I'm moving my hand right now. These are how my like the fingers are grasping the steering and things like that. And so those memories are the non-declarative memories. There are also habits and emotions that come under non-declarative memory, right? So there are these different types of memory. I like this way of categorization because this is also showing the information flow in the brain, of how the information flows, right? Every memory, even the long-term memory has to go through a sensory memory and a short-term memory, and then it will be converted into a long-term memory. And at every stage, the brain assesses by different mechanisms whether this information is important or not important. And if it is important, it stores it as a long-term memory. If it is not, it's discarded. So these are the different types of memory. All right. Scientists have also identified now that there are different brain regions involving in these different types in encoding these different memories. For example, we have identified specific brain regions. Now, this is from the outside of the brain that you're seeing, specific brain regions that are encoding sensory information or are responsible for the sensory memory, like the touch, the taste, the vision, hearing, and the smell. And moreover, then they have identified how the information from the sensory systems then go to this front part of the brain called the prefrontal cortex. And this is the one that's responsible for the working memory, where all these interesting behavioral paradigms are designed. And then for the long-term memory, we have other limbic regions. Part of it is hippocampus, which is responsible for episodic memory, or the memory that is responsible for telling the episodes of our lives, right? Then we have amygdala for fear memory or emotional memory. And we have cerebellum for motor memory. And this is what gets disrupted in neurological problems like Parkinson's. Now, I want to explain why and how do scientists how do scientists figure out that these different brain regions are responsible for these different types of memory? Like, how did we come to this conclusion? And one of the cool things that we do in science is to really understand the function. We just either delete it or disrupt that region and see what kind of functional loss we have. And just by this strategy, we can start to understand the specific memories. So I'll give you two examples of how early on, or what two examples that really gave us the initial information about how specific brain regions can be involved in these different memories. The first very famous example comes from this example patient called Phineas Gage. Now, Phineas Gage was a railroad worker, and he one day, I think when he was 25 years old, he was working with his team and trying to making a railroad bed. He was trying to put some explosive in one of the holes with a big damping iron rod. So he had like this huge iron rod that you can still see in his picture. And while he was putting that explosive just burst and literally went through his left cheekbone, as you can see in this schematic, and came out of the skull and went 80 feet away and landed. And after 10-15 minutes, Phineas Gage just stood up, went to his teammates and said, actually, I think I need to go to the doctor. I think there is some blood loss. And he literally walked and he was fine. And the doctors initially didn't believe that an iron rod literally went through your brain and you are still surviving. And it still is a miracle case. So the people did surgery on him and he survived and he lived many years after this accident. Scientists later understood that during this accident, his frontal lobe, or this front part of the brain, was actually completely damaged. And what happened was, after the accident, Phineas Gage's personality changed. So before the accident, Phineas Gage was a rational, a very nice, nice to everyone, and he didn't used to drink alcohol and all that. But after the accident, he became really reckless. He could not plan things. He was very irrational. He could not keep up to any of his job. He became an alcoholic, and he became like a very antisocial person. And they realized that maybe this frontal lobe is really responsible for helping us all maintaining our personality, or helping us with all these higher order things, including working memory. And so this was, I think, one of the first cases in medical history that really helped us to tell a function of a specific brain region. The other example, which is a much more popular example, it's also relatively recent, comes from this guy called Henry Gustav Molaison. Now, Henry, when he was 9 years old, I think he's from Connecticut, and he was 9 years old, he went through a bike accident. And following his bike accident, he started to have all like these big seizures and epileptic seizures, and they were very disturbing for him. He would get these fits or seizures. And initially the doctors tried to curb these seizures using some drugs or pharmacological methods, but it didn't really work, and his seizures got worse and worse. And so when he was 27 or 28, they decided to remove the brain region that was the locus of these seizures. And so they removed the temporal lobes, which included this hippocampus. Okay, and this was one of a kind surgery. This was, I think, done in 1950s. After the surgery, Henry survived the surgery, and after the surgery, his seizures went away. But what he experienced is something called anterograde amnesia, and that is that he could not make new memories. His previous memories, his already existing memories, were all fine. He still remembered how he grew up. He still remembered everything till before the surgery. But after the surgery, he had absolutely he had no capacity to form new memories. And this really helped us to start the research in memory, where we started to understand the function of hippocampus into memory formation. For many decades, scientists studied him and develop a lot of behavioral tasks around him to understand what kind of different memories are affected. One of the funny examples is that is his friend would go and meet him, and he will always say hello, Henry, and then he will slap him and then leave the room. And then 15 minutes later, he will come again and he will slap him, and Henry will really respond and greet him like he has never met him before this. And he will come again and he'll slap him, and he'll do this multiple times, and Henry would have no memory that he was just slapped by this guy. He would still greet him very nicely. After 2 or 3 days, Henry would remember or have the sensation or memory of the pain that he has been slapped multiple times, but he'll have no memory of how he caught the pain. So, this and many such interesting tests were done on Henry, and he was, I think, one of the reasons why we have such huge advancements in memory research. All right, so this is how early on the studies were done using interesting behavioral tests and studying these interesting patients. But now we have developed some other tools, some other more advanced tools of looking at brain activity, right? For example, we know functional magnetic resonance imaging can help us to look at different brain connectivities. There is diffusion tensor imaging or electroencephalogram, which can record the electrical activity of different brain regions. And now we have much better information about how specific brain regions get activated and how they get activated together or in different times during specific memory formation. Moreover, scientists appreciated the fact that some of these memory mechanisms are actually conserved across species. And they use several different animal models, some that were invertebrates like the C. elegans, flies, or Aplysia, some that were vertebrates but lower like the zebra finches or zebra fishes or frog, and many mammalian models. they use some more invasive and elaborative tools to understand the mechanisms of memory. And so today, a lot of the principles that I'll tell you about how the neuronal communication takes place and how memories are formed will come from the studies that were done in a lot of these mammalian systems. So this brings me to the end of my part one. And now let's understand how does neuronal communication takes place. Now, and why it is important to understand memory. Now, I want to give you guys a hypothetical scenario. Intuitively, it makes sense that every memory is stored in the neurons or the cells in the brain really stores information. Now, one thing about the brain is we are limited by the fact that the number of cells that we are born with, we live with those number of cells throughout our life, right? And this is in contrast to the cells in the cells that are in the blood, which gets regenerated on the skin cells that keep regenerating every few days, right? And so if the information is stored in the cells, do you think that limits our capacity to store information? Let's understand through a hypothetical scenario. Let's imagine if the neurons, 100 neurons, store one packet of information. And we have five major senses, vision, auditory, taste, smell, and touch. And we receive two packets of information that are important per sense every hour. We receive millions of information every second, but maybe two packets of information are important every hour. So every hour, if we have 10 packets of information from these five senses, we are exhausting 1,000 neurons that will contain those information. In 24 hours, we'll exhaust 24,000 neurons, and in a year, we'll exhaust 8.7 million neurons. Now, if that is the case, how many years would it take to exhaust 100 million neurons that we think is there in the brain? Can anyone tell me? Can you think about a number? The number will come somewhere around 11 years. So 100 million neurons, if all of them are storing information, will get exhausted in 11 years. Is it the case though? That's not the case, right? Because we still maintain the capacity to learn throughout life. We still, even when we are 60, we can still learn a new language or a new instrument. And how that is the case? If the neurons are really storing the information, then how do we get almost infinite capacity? Well, the answer lies in the, the answer lies in the neuronal connections. And I'll explain you why in this section. So we know that brain contains a lot of different neurons. This is from a brain region called the visual cortex, and you can see so many different neurons. And if you zoom in further, you can see a particular type of neuron called the pyramidal neuron. And these neurons have very specialized structure, and these little projections are the dendrites. And if you zoom in further, are the dendritic spine, as a similar image that I showed in the title slide. And these thorny projections are called the dendritic spines. Now, we have way more connections than the number of cells. So although we may have around 100 million neurons, and each neuron can make 1,000 connections with many other cells, and we have close to 1 trillion connections in the brain. And so I'll tell you how these connections actually, and these connections called synapses, are actually important for memory storage. But before that, let's take a historical perspective and understand a little bit more about what neurons are. Now, historically, we didn't really know that brain comprises of these independent or individual functional units of life, or functional units of processing called neurons. In one of the most famous arguments was between these founders of neuroscience called between Camillo Golgi and Santiago Ramon y Cajal. And Camillo Golgi at the time, back in 1870s, he was a very famous scientist. He developed his technique of Golgi stain, and he was the one who came up with the idea that brain is nothing but a mesh, and most of the information processing takes place between the different network of meshes, and there is no independent unit in the brain. Santiago Ramon y Cajal, interestingly, using the same technique that Camilo Golgi used, he came up with the idea that actually there are independent units in the brain, which are called the neurons. So he came up with the neuronal doctrine, and they shared the Nobel Prize. But till the day they got the Nobel Prize, they kept with their interpretation of what they looked at in different brain sections from many different species. And now today we know that Ramon y Cajal was actually right, and there are these independent functional unit of neurons in the brain. Now, neurons, we know, have these specialized structures, and they are special in a way that they are electrically excitable. Now, I know that most of you might have heard some of it from Andre's talk, so I'll try to go here a little bit faster. So the neuron has a part called dendrite, which receives information from the upstream neurons. It has a cell body, which processes information, and if it decides that information is important, it then sends the information down to downstream neurons via these axons. And like I mentioned, neurons communicate via electrical signals. So this is just an example of if we record using this recording electrode, if we record from the neurons, we see that during neuronal communication, there could be this generation of millisecond long electrical signals called action potentials, and they are the ones that help in communicating an information from one cell to the other. Now, the electrical nature of neuron is also historically very, very interesting. And how did we come to understand that neurons? Now it makes all intuitive sense, but historically, scientists had a lot of arguments and a lot of different theories before we learned that neurons can be electrical. So it has an interesting history that I want to briefly touch upon. It started with Isaac Newton, that I assume everyone knows here. Isaac Newton, in addition to discovering gravity, really also theorized that the information sent to our peripheral parts of the body is electrical in nature. So there is communication in the biological systems is electrical. He didn't do any experiments, but then Luigi Galvani, again a very famous scientist known for other reasons, he was the first one to develop a battery. But he was originally an anatomist, and he electrically showed for the first time that the communication is electrical in nature. And his experiments are famously called the dancing frog leg, which you can all search on Google. What he did was he dissected out a frog, dissected out frog legs. And sometimes the frog legs would have a nerve terminal that if he electrically then stimulated, the frog leg would move. It's isolated from the body, but it's still responding to the electric signals. And he would do a bunch of cool experiments where he would put like bunch of dissected out frog legs outside in the balcony and wait for the lightning to strike. And as soon as the lightning strikes, all the frog legs would jump. And so he would do all these interesting experiments. And he, for the first time, experimentally showed that the nervous system is electrical. Giovanni Aldini he was a crazier person. He did some similar experiments in human corpses, which I don't think in today's world will be ethical. Julius Bernstein, for the first time, described the concept of action potential, how this information has to be a really fast millisecond long signal. And Alan Hodgkin, Andrew Huxley, and Bernard Katz were one of the first people to see actual action, to record an actual action potential and develop these electrophysiological tools to see an action potential. And they studied it in another interesting animal model of squid. And squids have really giant axons. And so they were able to record from these axons the action potentials. And these techniques are still, the principles of these techniques are still used today. I still use them every day in my experiments. And so for me, they are the rock stars of neuroscience. All right, so this is the brief history, which I thought was very interesting. And today we can do electrical recordings in brain slices. We make, for example, now more commonly in our labs, we use rodent models, mice or rats, and we make different brain sections. This is an example of a hippocampal section. It's a coronal section, very thin section. And if you zoom it in using a microscope, You can see individual cells, and you can really put in an electrode in the cells and record different types of current. The currents that really contribute to generation of action potentials, and the currents that are coming in due to a communication with other neurons. And similar electrical recordings can be done with a bunch of different cells as well together, or getting an average response. Okay, so we learned about the electrical communication between the neurons, and where do these neurons make the electrical communication? They make at these specific sites called synapses. And this is where the two neurons come close together and communicate. So now let's understand the structure of a synapse a little bit more. Now, synapse can contain contains there are two components. There is a presynaptic component, which is a part of the axonal terminal, which is this part, and this contains synaptic vesicles. These circles are the synaptic vesicles, which contain chemicals called neurotransmitters that get released when the action potentials come down to the axon. And then there is a postsynaptic component. This is a schematic of a dendritic spine. And this contains a number of receptors, which basically gets bound by the neurotransmitters and open up. They undergo conformational change, open up and cause an influx of ions that will then depolarize a cell. Now, the strength of the synapse is defined when a synapse is activated and how likely it is to generate an action potential. The stronger the synapse, the more likely that the release of the chemicals from that synapse will generate an action potential. Cool. So the neuroscientists are not really imaginative people. We didn't really imagine and made these drawings. This is how a synapse looks like in real as well. This is an evidence of that. This is a high resolution image of a synapse that is acquired using electron microscopy. And here you can see that this is the dendritic part. And you can imagine that this is the dendrite that's literally coming out of your screen. So it's basically that section. And with the neck, you can see this spine. This is a dendritic spine, and it has a presynaptic partner, which has these dots called the synaptic vesicles. And this dark portion is where all the receptors are. This is where the chemical gets released, and the receptors open up and cause the depolarization or ion influx in the cell. Cool. So now comes the part. Now, how do these connections, why have I spent so much time talking about the synapses and how synapses are important? The cool phenomena is that these connections are dynamic. These synapses are plastic in nature. They change with experience. If you're learning a new information, these connections strength will increase. And if you are if there is a memory loss, these connection strength will weaken. And this is an example of how the connection strength increase can be observed. This is from a real scientific data, where you can, if you focus on the spine, you'll see that the spine volume will increase. And this is in real time telling us about this phenomena of synaptic plasticity. And this is the cellular mechanism of how cellular mechanism of memory formation. And the question that I asked in the beginning, the answer lies in the connections, because these connections are dynamic and they change with experience. Okay, so let's go time. Oh yeah, it's 7:45. Let's go a little bit faster. So let's assume. So now let's see how the principle of how the neurons or the synapses can increase the strength. One mechanism could be this that I'm explaining here. So imagine our neuron A has 3 different connections from different neurons, neuron 1, neuron 2, neuron 3. Now, if you're playing a piano and you are trying to learn a specific skill while playing a piano, you will activate certain neuron. And in this example, let's assume that neuron 2 gets fired again and again. So now you practice and you improve a skill of playing piano. The neuron 2 will get activated again and again. And most likely, if the neuron 2 fires more than neuron 1 and 3, the synaptic strength of that neuron will increase. And the synaptic strength will increase means that when the neuron 2 fires, it is more likely to produce an action potential in our cell, in our neuron A. And this is the concept that was theorized long time back, which is the neurons that fire together, wire together. And it was developed by this physiologist Donald Hebb back in 1949. And this is again reflecting the fact that the connections that get strengthened are more likely to generate an action potential in the neuron of interest. So the neurons that really fire together will wire together for memory formation. And let me actually skip this slide for the time constraint. We go to this. So one way of really activating synapses is by repeated stimulation, repeated electrical stimulation, like I mentioned. So if you keep doing things again and again, you'll repeatedly stimulate it and activate synapses. But that cannot be the only way of learning, right? Because there are certain things that you might have to learn in a more in faster fashion. Like in one trial, for example, if you look at a tiger, you need to sense, you need to learn that that's a danger signal. In one trial, you cannot look at the tiger again and again to really learn that it's a danger signal. So there are more mechanisms to really cause the synaptic plasticity. There are more than one mechanisms of instead of just repeated firing. There can be neuromodulators or chemicals that can get released in the brain and that can modulate the synaptic strength. And we might have heard of some of it. For example, we might have heard of serotonin or dopamine or acetylcholine that can get released from specific brain region and can really affect or modulate synaptic strength during different behavioral paradigms or different learning paradigms. There are also other neuromodulators like endocannabinoids, caffeine that we take almost on daily basis, and neuropeptides that can get released within the brain region and can modulate synaptic strength. And so there are these different mechanisms that increase or modulate the synaptic mechanisms. But now the thing that we want to understand is how do these synaptic strengthening takes place during specific memory formation. So we are still trying to figure out specific mechanisms during synaptic during memory formation. All right, so let's come to the third part for the sake of time, because I want to touch upon this. This is kind of the thing that really connects us to of the synaptic strengthening to the memory formation. And this will talk about information processing at the synapses. So one of the conundrums so far that if you guys are catching up with me, I know I'm going a little bit faster, but one of the conundrums is that the electrical responses are millisecond wrong, right? And how do they get converted into the synaptic changes, which is responsible for the memory formation that lasts for minutes to many, many years, right? So this millisecond signal is how the neurons communicate. And even if it's important information or not, the signal is always going to be electrical. How does it get converted into a long-lasting change in the synapse? And for that, we give, and what's the reason? And for that, we give credit to Eric Kandel, who's still a very active scientist. And he showed us, gave us the first evidence of the mechanism of memory formation by studying sea slugs or Aplysia. And in Aplysia, he used two different types of learning paradigms. He used habituation. So if he gave a tactile stimulus to the Gill-Siphon, which is a part of the part of Aplysia, if you give a tactile stimulus by just touching a pencil or something, the Gill-Siphon would retract. But if you give it again and again, the retraction would become less and less. It's similar to how we get habituated by living in a small, in a loud city, for example, right? If we instead go to a quieter place and then come back to the loud city, we suddenly feel that, oh, the city is pretty loud, but then we eventually get habituated. So similar habituation process happens with the Gill-Siphon reflex here with the tactile stimulus. And the other paradigm he studied was sensitization, where he gave a tail shock in addition to the tactile stimulus. Now, tail shock was a very dangerous signal, and there was a huge retraction of the gill and the siphon. And what he found was that similar to the repeated mechanism or repeated firing that we see in the neurons, if you can keep on giving the tail shocks for many days across the training session, the duration of the withdrawal would be way higher. So as compared to the single tail shock, which you see in this light blue, if you give 4 trains of tail shocks, it will go really high. Now, using these learning paradigms, um, Dr. Kendall not only figured out the neurobiological basis of this, found out important neurons and the mechanism of learning at the neural level. He also found out that the molecular basis. So what he found out was serotonin, which is a neuromodulator that was getting released during these behaviors, activate specific proteins for either a short-term memory or habituation kind of phenomena, or a long-term memory, leading to more different protein like a Krebs activation, which would cause gene expression changes. And this was the beginning point of us neuroscientists to really understand or really figure out the proteins that can alter during proteins that get transduced from the electrical signal, and they can get lasted longer to see an increase in the synaptic strength and eventually lead to memory formation. Now today, we know thousands of protein that underlie synaptic plasticity or changes in this modulation of the synaptic strength. I don't want to really go into the details of each of these proteins, but I just want you to appreciate that we have identified so much biochemical signaling, starting from like one pKa molecule or Krebs molecule to like thousands of proteins that can modulate the synaptic strength. And to really summarize decades of research in a simple fashion, I can say that the way I try to explain how the memories are formed from the synaptic level is that you have an electrical stimulus, and then you have some secondary messengers that get released, which last for 5 to 7 seconds. And this can be either calcium or it can be cyclic AMP. And that leads to activation of biochemical signaling that lasts for 1 to 100 minutes. And depending on the type of biochemical signaling that gets activated, you can have long-lasting changes in the genes, and then the gene expression profile changes, which causes specific changes in the synapses. And these changes can last for many hours to up to many years. And so that's how the electrical stimulus, which is a millisecond long, gets transduced into changes that are long-lasting. Now, there are many different, so these biochemical signaling, again, like I mentioned, are conserved across different brain regions and different types of memories, and also across different species, suggesting the robustness of information processing mechanisms. And over the years, these proteins have been studied in many different ways. I can just quickly go over. In 1950s, for example, the protein function was studied using pharmacological blocking of these protein activity using specific drugs. In the 1990s, we started to understand the genome a little bit more, and we started deleting these genes from the genome of specific animal models. And in the 2000s, we started actually visualizing protein activity during synaptic plasticity. And so I want to focus a little bit more on that because that's what our lab specializes in. This is the last part. These are the, so we developed something called biosensors to visualize protein activity, and they are based on the conformational change that occurs in the protein. And what we do is, in very simple terms, we attach different fluorophore pairs in the proteins, and we then detect how close or how far these protein or these fluorophores are during the conformational change. And just want to end with a beautiful image or video of activation of a particular protein. For example, in this case, CAMK2, in response to a signal that causes synaptic plasticity. So if you focus on this spine, you'll see the change in the color, showing how in a synapse-specific manner, this CAMK2 gets activated, and which is also locked to the stimulus. And you can see the spine then after that increases in volume, suggesting an increase in the synaptic strength. There are some other proteins that get activated in a more global fashion. So if you see in this spine, when we give the stimulus, you'll see the change in the fluorescence and that is a more global activation. This is an example of another protein called ROA. And similar to these sensors, we have developed a dozen sensors now. Rohei Yasuda, who's my mentor, has pioneered in developing sensors, and that has started revealing us a protein activity at single synapses during the synaptic plasticity. Some of them can be spreading signals, or some of them can be synapse-specific signals. And so now the future direction, the thing that lacks is that we have the biochemical computation information, we have all these biochemical signaling, we know that synaptic plasticity takes place, and there are many mechanisms of synaptic plasticity, either by release of neuromodulators or release of persistent activity of a particular neuron. The future direction is to really link it to the actual memory formation. These are like we have developed the theory, but the future direction is to now take a behavior, take a memory, and then understand the specific activity dependent synaptic plasticity mechanisms that happen, and what specific biochemical signaling that activates during this memory formation. And understanding these proteins or these biochemical signaling will bring us back to our original problem of really solving memory loss, right? So using or having more understanding about these proteins will help us to develop more effective therapeutic strategies to correct for synaptic plasticity loss, right? And so this is where the scientists are heading as well. They're already planning on designing drugs to target these synaptic proteins in a brain-specific fashion. All right, this is the end of the talk. This is the team at MPFI that I work with. It's the whole Yasuda lab. It's a huge group, and these are all really talented scientists, and it's been a pleasure to work with them. And thanks.
Oh, fantastic, Anant. So on behalf of everybody, we'll give you virtual grand applause of hands for fantastic presentation. And just as a reminder to everybody who is participating, if you have any questions, this would be the time now to type them into the chat window. And to give you some time to do this, I will kick it off with a couple of questions for Anant. So the first one is, so if you look forward to something, you know, like an experience, does it have an influence on memory formation? Contrast with if you are not looking forward to do something. Do you guys know anything, whether there is a relationship?
Very good question. And we have actually done a bit of research on the motivational state of the, like using these rodent models. We have found the motivational state really determines the learning of a particular task. So for example, if you have a reward at the end of a task and the animal knows that there is a reward, there is a high motivational state to learn that information, right? And so the animal will perform way better if it is highly motivated. And so of course there is a huge component of that.
So do you think that translates to the human experience? Let's say you're looking forward to a concert or something. Does that dispose you in a way that you're more likely to make good memories or strong memories compared to you have to go to the dentist, let's say?
Very good point. We think that some of these principles do transgress to how humans form memories as well, and definitely there is this element of motivation which really interferes with your memory formation for sure. We actually, I'm not aware of any of the fMRI data, but there are behavioral tasks that are developed based on reward-based behavioral tasks developed, and so indeed it is true. And not only motivation, there are several factors of how memories can be affected. For example, I'll give you a small interesting example. Stress, if you are in a stress situation, the learning gets affected, and so intuitively you can imagine that in a stressed brain, you will not be able to learn well. And so this is also very extensively studied in neuroscience, yeah.
So I hope there's still some students coming up with a question, but while we are waiting or giving them a few minutes, Let me expand on this. Do you, knowing about how memory is formed, do you think that we can actively influence in how good we are at making or memorizing things if we understand the mechanism, or is our understanding of the mechanism totally disconnected from the actual formation of or our ability to form memories?
So if I understand the question correctly, you are hinting to the fact that us trying to understand the mechanism actually helps us improve the memory, or the understanding is kind of unrelated. I think the motivation so far, honestly speaking, has not been to really manipulate the human memory. So far, we are still at a stage to really understand the basic elements of the memory, and we are still our techniques have still reached far enough to understand memories in rodents, right? And in their systems, the memories are still simple, and we can only take a few principles of that, but that might not be directly relatable of at all times to our human memory, and our human memory may be more complex. So I'm not sure we are at a stage to really start improving memory, but we are still at a young stage of understanding.
Oh, fair enough. I have a question here from Spencer.
Yeah.
Is there a sense what's most likely to become a long-term memory?
Sorry.
Yeah, he's asking whether there's a sense. He said that is the most likely to become a long-term memory. I interpret
Yeah,
this to say.
So it doesn't really depend on the sense, but it depends on the type of information that you get from a particular sense, right? And so I did talk about the three main senses that humans use. The sense of vision. Vision is one of the most prominent, I think, sense that humans learn the most with. The auditory information, and the information of touch. And so these are the three senses that humans use a lot to learn. And so most likely information coming from these senses will be converted into long-term information rather than information coming through odors or through taste, for example. But also integration of different information, integration of information from coming from different senses also helps the brain to understand that it's important. For example, if you are preparing for an exam, and if you read it, if you hear the lecture, if you read the lecture, and if you have different ways, if you ask the friend to tell you back some components of the lecture, so you are having the same information coming in different ways, and that helps the brain to decide that, okay, this is an important information, I should probably remember it, rather than just coming in through one sense, which is through vision, for example.
Okay, next question from Como. Why do humans have vivid, specific memories of bad experiences that are stronger, in parentheses, than other good memories?
That's a great question. I don't really have a direct answer to this. What I think is the negative memories, like if you're embarrassed in a class, for example, if you're bullied in your childhood, that gets combined with an emotional component. And these emotions, when you have like a fear or a stressful emotion or a stressful experience, there are these specific neuromodulators that we know today gets released, like norepinephrine, and the brain is able to associate that kind of information more strongly with the emotions than many positive experiences. So that's a great question. I mean, this is again the theories that we have built. We still don't really have a direct evidence suggesting the same.
Okay, then Mia has a question for you. If someone has a really good memory, does that mean that their neurons are more efficient than other people's?
That's a good question. So this is something that I kind of skipped. So having a specific or appropriate number of synapses is actually important and important for the memory development. or memory formation. So there are cases where you can have many more number of synapses or many more number of connection, but that does not mean the brain is efficient. And if you have less synapses, again, it is not efficient. So having appropriate number of synapses are really like there is an efficiency component, very small efficiency component to this efficient memory formation. Yeah.
Okay, then we have a question from Lillian. If somebody has a very tragic experience, why does that memory tend to have a longer effect on someone than a good memory?
This is again going back to the previous explanation that I gave of the negative memories. The tragic experience again have a stronger emotional component, which is easily associated by the brain or the working memory component when it is trying to associate this memory. And this is again a part of the post-traumatic stress disorder, right? And this memory tends to have a longer effect than a good memory. This is similar explanation to the previous one. Yeah.
Okay, you have a little longer question from Doug. He writes, Matteo Ricci was an Italian Jesuit in China who became a memory coach for Chinese students wishing to do well on examinations. He advocated imagining a memory palace or imaginary house that you associate with items to remember. Is this accurate that visual imagination is associated with long-term memory? Any other ideas your research has uncovered for remembering better? Any tips for students?
No, this is I'm not aware of Matteo Ricci's work specifically, but this is true. Like I mentioned before, that vision is one of the stronger senses in humans that helps us to learn and form memories. But one of the ideas, like I discussed before, is how you can combine different sensory information while you are learning new things. You can use either an audiobook and you can read it, and you can see a presentation of your teachers, and you can get the information by many different ways. And combining the same information by different senses will help you to remember things better. Another, I mean, there are all these other things. We have a circadian component of learning. So I don't know if it is generally true, but it is believed that some of these proteins that are important for synaptic plasticity are activated more in the morning than in the evening. That's why I started with the brains being tired a little bit. So learning in the morning is also more helpful.
Okay, so another question from Spencer. What is instinct in this contest? Does instinct play a role? I'm not exactly sure what he means, but
yeah, I'm not sure either.
Maybe Spencer, if you're still there, you can expand on your question. And while you do that, I will ask Anant another question. Do we have any understanding why our memories are often false the longer we are past the event? And people still believe firmly that what they are saying is true, but objectively it isn't true. But somehow memory doesn't seem to be stable. It seems to be very fluid in the sense that
very malleable. Yes.
So any explanation from the research you guys are doing?
So again, the research is at the level of behavioral tests. The memory definitely is malleable. We have identified certain factors that really interfere with your memory recalling. So for example, if you describe about your 25th anniversary, right, and you tell that I did this, we went to this restaurant, blah blah, but you might have gone to that restaurant again in some other instance, and you will see that over the period of years you'll start mixing up both those events. And so one of the interference factor could be things that are very close to the memory that you are trying to remember. So you try to you learn about some other information which is very overlapping and you have a memory which is very close to the memory. So those are some of the interference factors that keep on getting added up, which makes the memory more and more malleable. And there are also other interfering factors. Again, some of these components can hint towards mental illnesses that makes the memory more malleable than during normal healthy aging.
Okay, then Ryan, and his question is also a little bit longer if you want to scroll down to the chat box yourself.
I can read it.
But I read it out loud for everybody. When considering the idea of reward-based motivation, does a hierarchy such as Maslow's hierarchy of needs tend to apply, as in, do you observe varied motivation based on need? Furthermore, is there a general way we gauge importance?
Yeah, again, that's a very, very interesting question. The motivation is always based on need. A lot of these behavioral tasks that we do in animals are done with the starvation protocol. So we starve the animals before we start the tasks, and the reward, if they complete a task successfully, will be the food at the end. So that's the motivation that they have. But yeah, the motivation is solely based on need, because we cannot have any higher order motivation studied objectively in lower order animal models like rats. So it's absolutely critical in our behavioral paradigms.
Okay, then Spencer expanded on his original question, what is instinct in this context, by saying, like an instinctual understanding, is this like a memory or is it something else entirely?
Yeah, these are some of the things, we are not there yet, to distinguish between the instincts. Like, it's not, I don't really have a logic for it. It's just my intuition or my instincts. It's an instinctual understanding. I'm not sure. I feel, again, this is a feeling. I don't really have a solid evidence for it, but I feel that this comes in from the prior knowledge or prior understanding that you might have, or you might have unconsciously gotten by listening to your elders or listening to your friends, for example. And you develop kind of an instinctual understanding unconsciously. So it can be considered as a non-declarative form of memory, which you don't consciously recall collecting. So it is, in my opinion, a form of memory.
Okay, very good. I don't have any other questions, so I can ask everybody to unmute and give a hand of applause to Anant for his great presentation. And I hope everybody enjoyed it and had a good time and learned something about memory formation. Anant, I think you're still there, right? You dropped off here from my screen, but hopefully
I'm still here. Yeah. Thank you so much again, Stefan. This was a pleasure. This was fun. Yeah, but if you guys have any other question, you guys can definitely reach out to me, and I'll be happy to answer questions.
That's very generous of you, and everybody can still see his email and also his Instagram account, so you connect with Anant in your most favorite way. On behalf of everybody, thank you very much again, Anant, for being so generous with your time and share some of the work you have been doing and some of the work other people have been doing that sort of shaped your path studying memory formation. And I'll thank everybody for tuning in. We'll have another lecture in February. We'll send out the details in the invitations. And until then, I wish everyone good night and thank you very much again.
Thank you. Have a good night, everyone.
Good night.